All right, everyone. We're going to get started here. Thank you all for coming. Thank you for those of you online. If you want, feel free to leave in the chat where you're joining us from. It is a super pleasure of mine to be able to have Dr. Ramesh Laungani here, who is a recent addition to the Hudson Valley, though originally is from the Long Island area, so this isn't crazy new. Ramesh has had kind of an interesting and hit a lot of different places in his career. So we went to graduate school at the University of Nebraska in Lincoln, and then was a professor for a while at Doane University. And then he probably could tell you a lot more about his decision to kind of transition and actually be, the chair of the science department in a high school in the New York City area. But then got inspired to get back into a university setting. And so Ramesh is one of the newest kind of biology, environmental science professors at Marist University, which is a big change now. And I'm so excited to have him in the area because we have definitely some similar interests, but this idea of kind of understanding nutrients, looking at grassland ecosystems, and also just being really creative with how he brings students and others into the research experience. And so I'm really pleased to have you in the area. I think this is always great when we have local people because hopefully this will be a great opportunity for you all to learn about his work, but also for us to share what we can offer here at Cary and be the start of a lot of new collaborations. So with that, I will pass it over to you, and excited to hear what you have to tell us.
Thanks so much, there we go. Thank you so much, Jane. So I start with a thank you slide because I always want to make sure that I give credit where credit is due. Whether I get to all the data is almost immaterial, but as scientists, it's important for us to recognize that we all do this work together. We're a community of scientists supporting each other. So I first want to thank Jane. Jane was one of the first people I met here when my partner and I moved up here, sort of hunting around for what does science mean in this area. And so immediately, beyond our shared interest in carbon and nitrogen cycling, I tossed this idea about, hey, Jane, can I bring my students up here and maybe they could do some sampling? And Jane, yeah, great, let's do it. So in the fall, it was my first set of classes at Marist, and so I was still learning where all the copy machines were on campus. And so I schlepped a group of 19 first-year students who had never been, many of whom didn't seem like had ever been outside before, and I said, hey, welcome to a prairie, and welcome to this plastic square called a quadrat, and let's figure out how to collect some data. So there's Jane explaining to my students what percent cover is. And so Jane, thank you so much for this, and thank you all for the invite. Hopefully I'll give you a little insight into how I think about science. I'm excited for the other conversations I'll have later today. And as Jane said, my hope is that this set of slides is really a mechanism for building collaboration. This data set is, or the data I'm going to show you today, is a small piece of all the craziness that goes on in my head about science and helping students get involved in science. And again, ultimately realizing that science is something that we build collaboratively. I think oftentimes students think of, for a whole suite of reasons, right? They grow up, they think about, the white-haired scientist in the basement working alone, and then they invent something, they meet their college professors, and they're in their office, and I think it's rare that students of any age see that all scientists are really, we're just curious kids, right? We're curious kids, and we can just ask ourselves, we're just good at articulating our curiosities. And so I was just chatting with Amy just before this about termites, and she could probably just tell from my face. I was like, I didn't realize this was what was going on about termites. I was a 5-year-old again, and so like I said, I hope this talk is really a springboard for collaboration. So I'm going to start off with some good news. 2024 was the hottest year on record. I was, in preparation for this talk, I was sort of clicking through the the videos of prior talks, and one of them that I clicked on talked about how I think it was a polar talk, said this talk has nothing explicitly about climate change. This is not going to be that talk, all right? Climate change is unfortunately everywhere, and it's really woven right through this talk. So very simply, right, the Earth is getting hotter. This is recent data from NASA. This is a recent headline. Unfortunately, I have to keep updating this slide with the new record that we are breaking in terms of global temperatures. And so, if any of you watch The Daily Show, this is a GIF this is a comedian there named Ronnie Chang. This is from a few years ago, but I think this is really telling. This is a temperature anomaly graph that even if you flip it upside down, the line is still going up. And in many ways, I thought this is actually one of the best explanations of what's happening to the globe in terms of temperature that I've ever seen. The science is irrefutable. There's no question about the human activity being the major driver of climate change, right? And so the globe is getting hotter, and so this is the title of my talk, right? That was on the website. And so I'll tell you about biochar. Maybe you've heard of it, maybe you haven't. But I'll talk about its impacts on plant communities and how I help my students understand that their science can be used to tackle, at least in part, this global problem. And that's really, teaching at Marist, we're primarily undergraduate institution, and so I see my science as really a gateway for students to know that they can access science, and that their science can take on global problems. So this is the official title of my talk. This is the unofficial title of my talk, and I'll try to answer this question today. So we're officially Marist University. We changed like 2 weeks ago to Marist University, so like that's the new Marist University symbol. I guess that's supposed to be a U and an M, but yeah, we're Marist University. I will probably call it Marist College. Oh God, this is being streamed. Anyway, my brand officer is gonna be like, Anyway, so I'm at Marist. I've been there now for 6 months, so things are going good. All the data you're going to see today has all been collected by undergraduates. And these are my former undergraduates at Doane University in Nebraska. And so I bring this up because, as I said, when thinking about global problems, there's plenty of data about younger generations and eco-anxiety around climate change. And so ultimately, how do we help them understand that science is a pathway to solve problems, right? I also ask them, I often tell my students, when I get into conversations about what do you do with climate change, how do you think about this? And they ask me, how do you stay hopeful? And I say, I stay hopeful because there's data to collect, very simply, right? There's data to collect, there are questions to ask and answer. And so I try to frame their science as an act of active hope around battling climate change. Because without that, I mean, graphs, ANOVAs, data is all fine, but if we can frame it as this is your actions are helping us solve this problem, I think it gives them a little more motivation to sort of dig in on the science. So, as I just showed you, right, this is data from NASA. The Earth is getting hotter, and as we know, why is the Earth getting hotter? Well, this is here's Mauna Loa carbon dioxide data. This is data from the Keeling curve, historical data, right? I just looked at this data yesterday. This is where we are at. We're at 427 parts per million of CO2 in the atmosphere, right? Very simply, CO2, it's a greenhouse gas. CO2 is going up. This is the major driver of modern climate change. And obviously, we are at unprecedented levels for human history. And I always point that out to students and to audiences because, when we think about climate change, yes, we want to save all of this amazing biodiversity. We want to save these beautiful spaces. But a lot of climate change, I tell my students, maybe this is the wrong place to say, the Earth will be fine. The Earth has been around for 4.5 billion years. So when we think about fighting climate change, it's really about how do we save us, right? And how do we think about these unprecedented levels of carbon and climate that we haven't seen before. So this is an adapted graphic from Skeptical Science, thinking about the broad, this is a really big picture thinking about the carbon cycle. So, you know, these trees and this land really represents the trees and land on the planet. There's oceans and aquatic ecosystems. And really, right, as we've all learned in 7th grade, plants are really good at sucking up carbon from the atmosphere through photosynthesis. But vegetation and land takes in carbon, it releases carbon. The oceans, and what we found is that those two fluxes, actually it's a net sink into the ground. Okay, so there's actually more carbon being taken in from the atmosphere than is being let out. We have a similar dynamic with the ocean. It's also a net sink, right? And so if we think about this, ultimately we should be seeing, if this was our whole system, we should be seeing atmospheric CO2 levels going down. Right? So we should see it going down, but we have this. We have this flux. We have this up arrow. And this is the one that is really, we don't have, for lack of a better phrase, we have balancing arrows here, we have balancing arrows here, we don't have a balancing down arrow here. Right? And we put up so much carbon dioxide that we are actually overwhelming the net sink component of these other fluxes. And so this now becomes a source, hence the data of rising carbon dioxide levels. Okay? And I often show this regardless of audience, but I show this to my students because I also want them to recognize that in many ways climate change is just understanding a number line. It's understanding a bank account. It's understanding carbon accounting. Where's the carbon going? Where is it going in? Where's it going out? There's plenty of complexity in there, but let's start here, right? And so what's really important, and this is the work that I'll be talking about today, is this down arrow. Now, we need to reduce our CO2 emissions into the atmosphere. This flux needs to become smaller. That's not a question. But we also need to understand how do we build and understand a downward balancing arrow in this context. And this is really the center of my work. Carbon storage and carbon sequestration. How do we understand that downward arrow? And why does this matter? Why does that question matter? Because it's our choices that help us build our future, right? So this is a graphic from the IPCC, or the latest IPCC report. These are the shared socioeconomic pathways. Depending on the choices we make in terms of our emissions, that is going to drive what global temperature rise we are going to see. And you know, I'm glossing over a lot of complexity in this graph, right? There's a lot of stuff packed into each one of these colored lines. But the real gain here is that this is up to us. This is also from the IPCC report. I thought this was one of the most powerful graphics that they actually included in the most recent IPCC report. So if you are familiar with climate stripes, basically it's this graphic that explains that the world is getting warmer relative to a baseline. The redder, the more orange it is, the hotter it's going to be. And it really puts it into context. So that was me. I was born in 1980. Obviously, this is just in the graphic, lined up really nicely. So that was me. And so when we think about it, our choices are what are going to drive what the kid born in 2020 is going to see. And again, I bring this up to my students. Our future depends on our choices. And so I use that again to also frame my science. So given all of that context, what are the central questions of my lab? One, I ask very simply, I'm trained as a plant ecologist, so when I walk around an ecosystem, I think about, although I think about carbon and nitrogen cycling, I really am a community ecologist that studies nutrient cycling. How do nitrogen dynamics in the soil impact who's going to win competition? You know, I can think about sort of whole forest ecosystems or whole grassland ecosystems, but I'm really interested in that fight between species at a fine scale. I just happen to think about those fights around nitrogen and carbon. So I have students that have worked on questions sort of broadly, what are the ecological impacts of climate change? But today, I'm really going to focus on what are the ecological impacts of climate mitigation strategies. And the strategy I'm going to tell you about, as I said, as was stated in my title, is this thing called biochar. And so if you're unfamiliar with biochar, I'll give you the quick crash course here, right? Right, we need to work on this downward arrow. Plants are really good at sucking up CO2 from the atmosphere. Great, they're doing all the work for us, but, and I'll explain in a few slides, that carbon gets sucked up, but it also can decompose and go right back into the atmosphere. But we can take that carbon that was put in plant tissues and turn it into this stuff called biochar. And there have been plenty of studies looking at, and this is what biochar looks like. I actually should have brought some with me to like hand out. You can smell it and touch it, but it's just, you know, just looks like burnt charcoal. That's what it looks like. And, you know, one of the reasons it's thought about from a carbon storage perspective is that you can take any organic material, old coffee grounds, sawdust, dead grass. I've seen studies using manure, but we'll just think about plant material for today. It's pyrolyzed, I shouldn't say burned, it's pyrolyzed at really, really high temperatures under low oxygen conditions. And ultimately, that pile of pyrolyzed charcoal-looking stuff is sort of just a pile of carbon that was once in the atmosphere. And once you take that pile of carbon and add it to the soil, that carbon, in many ways, is just stuck there. It's stuck there. It doesn't decompose, or it decomposes very, very, very slowly from our best estimates. And so when thinking about biochar, there's been work suggesting that it is one of the many ways. It is not, like any climate solution, it is not the silver bullet, right? Biochar is one of many tools that we need to be using or could be using, that we need to be pulling out of our toolbox to fight global climate change. And so this is typically this is an oversimplified carbon cycle. You have a plant, it's full of carbon that's been sucked out of the atmosphere. It dies, it decomposes, and about 90% of that carbon, the vast majority of that carbon goes back up to the atmosphere. And so while that tree may live for a while, depending on what ecosystem you're in, I know there are a number of you that work in the tropics, this is the thing that made me nervous about actually speaking at Cary is that there are people working in so many places. I want you to know even now, I'm like, what am I saying wrong right now about these dynamics that someone's gonna be like, not in my system. But for the most part, this tree is gonna decompose and most of its carbon is gonna go back up to the atmosphere. And so while trees are really great carbon sinks, how do we store things on even longer time scales? So biochar can sort of short circuit that a little bit. So we can take that same tree, we do that same biomass, run it through pyrolysis, again, heat it under low oxygen conditions, turn about half of that carbon, half of that biomass into biochar. Now, very simply, you put organic material under high temperatures. We've all probably been near a campfire. The wood you put in the campfire is not the same wood you pull out of the campfire, right? So you're going to lose 50% of that carbon. But now, the way I think about it is you're going to gain 50% of that carbon in a more permanent form. Okay? And so we can then take that biochar, add it to soils, and there's work in all of these places that it has impact on all of these aspects of ecology. And so oftentimes when I think about biochar, I'm trying to explain this to my students, like, what is it? I show them biochar, they're like, who cares? What is this? I'm trying to understand this. And I often relate it to a pharmaceutical ad. And so we've all seen pharmaceutical ads, take the pill, right? There's some dancing and singing and there's dogs jumping through rainbows or whatever it is, right? Take this pill, it'll solve this problem, and then there's 5 minutes of side effects at the end of this where more dogs are jumping through rainbows and all of the things, right? Distracting you from the actual side effects. And that's really what I think about biochar. Biochar really is, it's a way to store carbon. So the questions I want my students to ask are, what are the side effects of this? Because if we understand the ecological dynamics and the impact of adding this stuff to soil, great, it's going to store carbon, sure. But will it encourage the growth of invasive species? Will it impact biodiversity? Could it stimulate more soil carbon loss? Right? Those are all side effects that we need to understand before we potentially deploy this tool at wide scale. And it affects all sorts of ecological dynamics. Jane and I were talking about how it could affect carbon and nitrogen cycling because of its physical structure. It holds on to ammonium in the soil really, really well, and so now you're changing fertility. That's going to obviously impact productivity and allocation and all of these things. And I'll show you some of that data today from work that I did with my undergrads. Excuse me. I'm sure that's great for the live stream. Coughing. Biochar has, like, the publication record of biochar. People have been getting interested in this in the last few years. This is work recently from Lee et al. Basically, all this is showing you is, much like the temperature, the amount of work on biochar is going up. People are interested in whether we should use it and how we can use it as a climate change mitigation strategy. And I have funding opportunities here largely when thinking about collaboration. How can we all, whether it's Marist and Carrie, how can we think about using this tool to help ask deeper and better questions? So this is what I'm going to focus on today. Here is essentially what I showed you before, right? That biochar is really good at storing carbon. This is the same figure. I've just sort of stretched this out. And what I'll get into here in a second is sort of the major questions that my students have asked. So, for example, one of the questions is, well, if we're going to add biochar to soil, how well does it actually sequester carbon in the soil? How well does it do it? Does adding biochar impact plant growth? How does it do it? And then, how does biochar impact biodiversity? Okay, how does it impact biodiversity? Ultimately, all of these side effects need to be understood if we're going to take this tree and turn it into this charcoal-like stuff to fight climate change. And so these are sort of the three areas I'm going to focus on today, right? And show you what my students have explored. So these are some questions. We'll be bouncing back to this slide quite a bit. So we'll start with the first question. Does biochar pyrolysis temperature impact carbon dioxide release from the soil? So as I mentioned earlier, you can take this, you can take organic material, you can pyrolyze it at a whole suite of temperatures, and you create biochar. Now, much like if any of you are bakers or have made pizza at home, depending on the temperature of your oven, it's going to change the quality of the bread coming out of the oven. Same thing with biochar. So what happens if we add biochar that's made at different temperatures? If we add that to the soil, how does the soil react to those different temperatures, temperature-made biochars? So this is work by Patrick. This was years ago. He did this for his senior thesis. All this work that you'll see today is largely done by seniors in college. And this was a simple question. Does biochar pyrolysis temperature impact soil CO2 release? And I always like to frame these questions as does. Yes, no. Does it? Because then we can ask the secondary question of, if it does, what is the effect? So what we did, we set up some soil incubations. We had jars without any biochar. We had jars made with used old coffee. So I went to the Starbucks down the street back in Nebraska, and I asked them for some old coffee grounds. I tried tweeting at Starbucks saying, hey, guess what? Fund my research, and you'll be helping fight climate change and teaching students. They did not pay attention to that tweet, unfortunately for them. But we made biochar at 400 degrees Celsius and biochar made at 500 degrees Celsius. We added that to soil, and we measured the flux of carbon dioxide coming out of that soil using sodium hydroxide. The reason I use this procedure, one, it was sort of a quick and dirty, on the cheap procedure, but Patrick was also a biology and chemistry major. So when you do this procedure, you have to do some Gen Chem 1 basic titration. If any of you want to pull that information, that trauma from the back of your brain, you might remember these long tubes with HCl, and you're dripping it in there, and you've got, like, you're waiting, did the thing turn pink or white or not? That's essentially what Patrick was doing to help us understand how much CO2 is coming out of these different soils. And so here's what we found. So before I show you the data, right, so he incubated these soils for about 2 months, a little more than 2 months, and we just measured the CO2 release. And so here's our first set of data. This is our control without biochar. Here's our sort of baseline. So, you know, I work at Marist. I teach undergraduate. Okay, so we have 2 choices here, 400-degree biochar and 500-degree biochar. I'm not going to show you. What do you think? I don't know, maybe this is not what's done at Cary. Maybe I'll just break the mold. What do you think? Which type of biochar is going to release more CO2, 400 or 500? Votes for 400? 400, 400, 400, 400. Any votes for 500? No, all right. All right, this is, Ashley's maybe, are you just like giving 500 a pity vote? Just like, I feel bad for 500, so like, all right, yeah, all right. Oh, that's some real fast consensus building. Either I'm in a room full of people who understand biochar dynamics really well, or I should have had everyone close their eyes and like put up a number, because maybe there was a whole bunch of like groupthink happening. Okay. So, right, we just need one person to say 400, and everyone's like, yeah, 400. That feels right. Right, so we had 400-degree biochar and 500-degree biochar, and okay. So 400-degree biochar might be releasing more CO2 than our unamended soils. So now the question is, where is the red 500-degree line? There's the red 500-degree line. So when we ran the statistics on this, we found that, in fact, 500-degree biochar was releasing overall less CO2 from our soils than our 400-degree biochar. And again, I bring this up, like this is a short-term, this is a short-term study, but I remember a conversation I had with Patrick after this. The data was great. He wrote this up in his little thesis. It was great. I was very proud of him. He gave a poster presentation. But the best part of this study for me was after that, right before he graduated, he said, hey, Ramesh, I wonder what would happen if we tried 600-degree biochar. That was the piece of data that was most impactful for me, because now I had hooked him, right? He was asking the next question, right? And that's why I think about biochar as a really great way for students to get into science, because it is something they can hold, it is something they can touch, it is something they can feel, and they feel that they are tackling this global problem. And so it's them asking the next question here that was really, really important for me. I couldn't put that data on a slide, other than a picture of me, like, and a single tear falling, other than me crying happy tears. So does biochar pyrolysis temperature impact CO2 release from soils? Yeah, it seems like there's evidence that it might do that. Okay, so does biochar impact the growth of woody plants? So this is work done by Maria and Tyler. And I highlight this. This was like the first time I published with my undergrads. Again, this is one of the things I'm most proud of, is that they were part of this. They went through the whole process with me, from start to finish. And so, does biochar impact made from different grasses impact tree growth? So what we did, we grew eastern red cedar, Juniperus virginiana, in biochar that was made from native grasses. Remember, I was out in the middle of Nebraska, so there was a lot of prairie around us. So we made biochar from native grasses. We made biochar from an invasive grass called Bromus inermis. I believe you have it here on site as well. There's a small patch over at the educational site. There's like a big patch of milkweed, and right next to it, there's a big patch of smooth brome that you can sit in, and when you sit in it, it's literally taller than, I guess, me sitting. So smooth brome is everywhere. And so I picked smooth brome because, one, it's ubiquitous, but also as we think about something like invasive species, what if we cut all those invasive grasses and now it actually turned into a way to store carbon, right? Can we take this bad thing and turn it into a good thing? We had native grass biochar. We also had native grass litter. As we had sort of three controls here. What if we didn't pyrolyze this stuff? How do plants react to this? And then we obviously had our unamended control. We measured eastern red cedar biomass, and we measured soil nitrogen levels. We weren't measuring any active rates of nitrogen production. We weren't measuring any net nitrogen mineralization. We were just taking snapshots of the nitrogen. And like I said, we published this work. So this is a really simple schematic of the experiment. We grew trees under these various conditions. And so, yeah, I mean, that's literally Maria setting up her pots. And here's what we found. When you add biochar to soils compared to dead leaf litter, you get more biomass. So here, I'll go out on a limb here because the work's already published. But if we add biochar to soils, we're storing that carbon that's in that biochar. And now, if we're making these trees grow a little faster, grow a little bigger, are we actually accelerating even more carbon removal from the atmosphere? Now, what's, I think, an interesting question, if you notice, these juniper plants basically produce the same amount of roots, but they seem to be producing way more stems with biochar compared to leaf litter. So that becomes the next question. How does biochar impact allocation, right? So where a plant is putting its carbon. Is it putting it more below ground? Is it putting it more above ground? So that's sort of the next question that I ultimately want some students at Marist to answer. But again, this becomes a nice little study. So the question becomes, all right, so we found this result. Why have we found this result? And I'm sort of spoiler alert here, right? So biochar impacts could be mediated through changes in soil nitrogen availability. So what you're going to see in a moment, so these are our various treatments. BB is brome biochar, native biochar, brome litter, and native litter. And then soil total inorganic nitrogen is what you're going to see. And you'll see some stacked bars in a second. So here's what we found. Our control pots without any biochar in it had significantly lower levels of inorganic nitrogen compared to our two biocharred pots, or biochar treatments, excuse me. And our native treatments, our litter treatments, had significantly lower levels of nitrogen. Now, this could be due to, leaf litter is more easily decomposable. You're going to get potentially more microbial nitrogen immobilization by that microbial community. What I found really interesting was the fact that there are no yellow bars on top of in this treatment. Like, what's going on with the ammonium? Okay, and I'm previewing this as if I've answered this question. I haven't. I don't know what happened with the ammonium. I genuinely don't. But clearly biochar is impacting the nitrogen dynamics. Is it not making, for example, is the ammonium that's in the soil inaccessible to nitrifiers? So the process of nitrification can't really happen? Well, maybe, maybe not. We still have, we have about the same amount of nitrate across here. So what's going on there? It's an open question. Like I said, my hope is to, as I get more settled at Marist, to have students actually asking nitrogen cycling questions, right? This is not really a cycling question. This is a what is happening with the nitrogen in a particular moment question. And so this is similar data, just in a different format. So this is an interaction plot. Everyone loves staring at these things. So let me orient everyone, right? So we're going to see 4 dots. The first one here is the total inorganic nitrogen levels that we found in our with bromus inermis litter. So essentially this treatment, okay? And then we had native litter, and so it'll be 4 dots. So here's what we found with just and that's sort of what you're seeing here, okay? So what we have a sense of is that the type of litter that you put in the soil is impacting the soil resource level. Not necessarily, like I'm not winning the Nobel Prize saying that we've done that, saying that that's the case. This actually, I think, is the most interesting result. And I know it's sort of mirrored here. But the fact that you have this species effect with litter, but then you turn different species into biochar, the litter of different species into biochar, and that species effect goes away. So species identity seems to no longer matter when you pyrolyze this material at, when you turn it into biochar. And, potentially this could have been driven by the fact that I was just using grass species, right? Native grass and invasive grass. Maybe they're, technically different. They have different stoichiometries, but maybe they're similar enough that in all reality, maybe we would see the line shift if I was using bromus inermis litter and sawdust. So that's one potential explanation, that you pyrolyze similar feedstock, similar starting material, and they're all sort of converging around almost like a single type of biochar. This is total inorganic N. We see the same sort of pattern with nitrate as well. Just to point out the axes here, we've got, this axis is going up to 10 milligrams of nitrogen per kilogram of soil, and now we're up to 6. So the rest of that difference is made up by our ammonium. But we're seeing, again, similar patterns, right? That species identity seems to be going away when you turn this grass material into biochar. And there's, like I said, there are my 2 undergrads. And what was really, again, I'll just highlight these sort of gratifying moments. I showed my undergrads this graph, and I said, do me a favor, figure this out. They figured it out. Like, yes, I had figured it out, but when I handed them this graph, they had never thought about an interaction plot before, which is not the most intuitive, right? 4 dots, there's sort of a line graph, but it's really sort of averages. I don't understand how to think about this, and they figured it out. They were like, oh, look, species identity seems to not matter. Gratifying moment. This is the next, like, Patrick, what's the next question? Oh, species identity doesn't seem to matter. That language from Maria's thesis went right into the paper. Okay, so does biochar impact, or can it impact, the growth of woody plants? Seems like the answer could be yes. And then, does biochar impact plant competition under climate change? So this is work, this is Cody Starman. That's him setting up his pots. And this is, at the end of Cody's work, I always like to tell my students that scientific research and writing it up is really storytelling. And scientists, as much as we like to tell ourselves how smart we are, we actually really just like picture books. And so when we are writing a paper, you got to think about the graphs that you want. What pictures do you want to draw? What's the storyboard that you want to set up when you're writing this paper? And so this is what we did. These were his data. We stood up on a table, and we were literally just sliding the images around with our feet, trying to figure out what's the storyboard of this result. I don't even know who took this photo, but someone just texted me this photo. Maybe it was one of my other students, because they were like, what the hell is my advisor and Cody doing standing on a lab bench? And so this work is in press literally like, what's today? Thursday? A week ago, I got the thumbs up that this work was accepted. So yeah, that's great. And so Cody's a co-author on it. And so what did we do? We grew 2 competing grasses, bromus inermis and perennial rye, with and without coffee, under elevated and ambient temperatures. This is sort of within the range of what we think is going to happen globally. 3 to 4 degrees Celsius between now and 2100. But we also, if we're going to understand competition between species, we have to understand, if we're going to understand interspecific competition, we also have to ask ourselves about intraspecific competition. And so we grew some monocultures of each species, and we measured plant biomass, and we measured the effect of competition by making a ratio of the growth of one of the species in interspecific pots over the growth of that same species in intraspecific, in monoculture. And I'll explain it a little more in a second. And again, so this is You can see the format of my slides here, right? This is sort of a common structure. So we had pots like this with coffee biochar under warming, without biochar under warming, with just some biochar, no ambient temperatures, and our sort of double control there, no biochar, no warming. Okay. So before we get into the actual data, we have to ask ourselves, how do we assess the effect and the impact of temperature on these interactions? So as I said, we made this ratio. And so just for argument's sake, let's think about this dark green species as our focal species. Okay? And so what we have is, we can measure the biomass of this dark green species when it's growing with a competitor. We can measure the biomass of that dark green species when it's growing without an interspecific competitor. And we can say, what's the ratio there? So if the ratio is approximately 1, so if we see a dot, if we get that measurement and that lines up with 1, we don't really have much interspecific. The dark green plant doesn't really care what the interspecific competitor is doing. So maybe that's allowing those two species to coexist, right? Maybe this dark green plant is like, sure, light green, sure, you do you, I'm good. I don't really care. We're not having, we're not really interacting that much. Or there's no negative impact. Because typically when we think about competition between species, we typically think of it as a negative interaction, right? Somebody's going to win, someone's going to lose, right? But in this case, if the ratio is 1, we have very little to no impact of interspecific competition, potentially leading to coexistence. If the dot is in this region, so we've got less dark green biomass in competition compared in interspecific competition compared to it in intraspecific competition. So we have let's just call it a ratio of 0.5. I'm just making that number up arbitrarily. Interspecific competition, right? These two are fighting much more than these two. Similarly, if we have a dot up here, if it's greater than 1, that means, these guys are really fighting and these guys are not fighting all that much. So these are this is sort of the outcome space that I'm going to show you. And what we have on our axes here is what's happening with these dynamics under ambient temperatures and elevated temperatures. Okay. Is that all making sense? I ask this question because thinking in ratios, I'm an ecosystem ecologist. I think about carbon and nitrogen ratios all the time, but thinking in ratios is just a weird thing. It's just a weird thing to think about, especially ratios that don't have a denominator, right? Ratios of 1, what does that mean, right? So then we have to, so I've shown you this. What's the impact of biochar? So how do we think about this? Well, in this is not real data. This is all sort of hypothetical scenario building. So we have maybe our community here, 2 species coexisting. Here's our RYP. I should go back, sorry. This is typically called as RYP. It's like a relative yield. P, I'm forgetting now off the top of my head, but relative yield of plant biomass. So the total biomass RYP for this dark species here, let's say we're at an RYP of 1 under current temperatures. These 2 species are coexisting. Maybe what will happen is under warmer temperatures, it's not so good for, like we have an RYP of less than 1, which means our dark species is being potentially outcompeted. So interspecific competition is having a negative impact on that dark species over here, sorry. So we go from 2 dark individuals to 1, and maybe we add biochar, and it exacerbates those negative impacts. That's one option, right? So we get deeper into that below 1 space. Okay. So if this is the result we see when we add biochar and temperature together, biochar is maybe not that great. So is carbon, but are we potentially losing biodiversity because of it? Okay, well, what's the other one? The boring result, right? We add biochar, and even if we have this sort of negative loss here, biochar isn't exacerbating that impact. Okay? And so then that becomes a different question. Do we want, what's going to happen to biodiversity under future climates? So here, biochar isn't really having an impact on those dynamics. What's interesting to think about is if biochar is having no effect on those dynamics, should we still put biochar out in the world because it's still storing carbon? Right? This is a boring result that might still be important. Right? A statistically insignificant result that might still be important to understand. And the third option, and there is a whole suite of options. You can imagine I'm just showing you a few here. The third option here is that you have this scenario and biochar actually helps bring the community back up. It brings that ratio back up to 1. So here, biochar is potentially making the negative impact of competition less severe and may promote coexistence, right, under future climates. So these are some options, these are some theoretical outcomes we can see. So those are our options. We've got red, nothing, and green. So we have the sort of dot moving down when we add biochar, we have the dot moving nowhere, we have the dot moving back up to 1. So here are some results. So without biochar, this is for Bromus inermis, for one of our focal species, but we saw similar effects on our perennial rye. We saw that warming significantly, the RYP was less than 1 under warmer conditions, potentially leading to competitive exclusion, potentially. This was, a short greenhouse study, so we're sort of making some extrapolations, right? Potentially leading to competitive exclusion. So then, but once we add biochar, that dot now moves up. We are now not significantly different from 1. And this is, again, a lesson that I tell my students all the time, as I just mentioned. Statistical significance does not mean, or statistical insignificance does not mean biologically unimportant. And so when Cody and I were working through this, I remember him seeing this, and he was like, oh, but the p-value is really high. It's a lot bigger than 0.05. I guess he literally was like, maybe I messed up. And I said, what do you think this means if biochar is getting us back to an RYP of 1? And eventually we worked through this, and he's like, oh, that means that species might actually be able to stick around. Oh, isn't that good for biodiversity? Now, the other wrinkle here is Bromus inermis, as you all know, is an invasive species. What do we do, right? Then it goes from the data to the should questions. What should we do, right? And those should questions are policy. Those should questions become management. But we need the data in order to make good, sound policy decisions, right? And so the question is, why is this happening? Why is this happening? And now we're into the full-on just conjecture part of things. So we measured biomass in these two species. And so we looked at the root-to-shoot ratio. These species are fighting with each other in the soil. These are fairly small pots, if you remember that picture of Cody with his headphones on. And what we find here is that when you just add biochar, and this is sort of the opposite of what we saw with that juniper tree. When you just add biochar, I should have blacked this out here, I should have covered these up. But when you just add biochar, you actually have more production of roots. This is in competition. This is in interspecific competition. These are in the interspecific competition pots. You have more allocation to roots. You almost have, not quite double, but you got a pretty big jump. So something's going on there. But the minute you put it under warmer temperatures, you have this very strange interaction happening. That difference seems to go away. And we see a similar pattern in both of our species here. What's going on? What is the biochar doing? Maybe this is the fact that we used coffee biochar. Maybe if we use sawdust, maybe if we use some other feedstock, maybe we'd see a different result. But again, Cody asked the next question when he saw this result. Hey, Ramesh, why are they allocating more roots in the beginning, under ambient temperatures, but not under elevated temperatures? Goal achieved, right? The next question was asked. And so, Here's some statistics. I can show you some p-values, right? I'm at Carey, so I felt like I have to show you at least some p-values here in a room full of scientists. So we saw that biochar increased root allocation, okay? But only in ambient temperatures. We saw a strange interaction, which we couldn't quite explain. We couldn't quite explain. Warming significantly lowered the root-to-shoot ratio. So, I mean, that in and of itself, why would a warming world, or why would a warming world change where a plant is putting its biomass? This has all sorts of implications for decomposition dynamics, carbon cycling. And so what we're seeing here is that in a warming world, whatever the effect of biochar is gets sort of mitigated under warmer temperatures. Right? The effect of biochar, which we see here, goes away under warmer temperatures. I'll just be perfectly frank. It's weird. I don't know. Still trying to figure it out. Still trying to think about it. But, you know, this was the graphical abstract of the paper. This was the thing, right? That we had this interaction where biochar and warming were interacting to potentially change competitive dynamics. And if it's going to change competitive dynamics between species, then we have to ask ourselves, what competitive interactions do we want to expose this biochar to? And I'll tell you, the reason I used this invasive species, if any of you have been out to the Midwest, the native species out there are all C4 plants, which is neither here nor there, but the native grasses are great, I love them, but they grow very slowly. They just take their sweet time growing. Invasive species, as the name suggests, grow really quickly, and so they work really well with undergrads who have to graduate in a year and do a senior thesis in an academic year. So just to be in perfect transparency, the reason I didn't have a native control here, or sort of a native species pair, was fully logistical, not for any particular reason, but I think it still begs the question, where, if we're going to do this, where should we be putting this stuff? What ecosystems and what competitive interactions do we want to actually try to potentially manage through the use of biochar? So, and Cody called it Project Dark Roast because it was coffee biochar. That's all Cody. That didn't make it into the final paper. I wish it did, but you know, science. So does it impact plant competition? Potentially. Seems like there might be something going on there. So when we think about biochar, you know, biochar has all of these potential impacts, right? It's climate policy. Where should we put it? Should we put it somewhere? Should we incentivize its use? We can quantify how much carbon is in that biochar. Should we be making some sort of carbon market around it? I'm not saying yes, no, but we can, right? That's a quantifiable thing. Should there be a policy around it? Can we think of it as a climate solution? Can we help people think about climate change differently through this thing that they can touch and feel? My students learned how to design experiments because of this burnt stuff. The questions about plant and microbial ecology are sort of endless, right? We didn't even get into who is in the soil and who is chomping or chewing or interacting with this charcoal stuff. We didn't touch any of it, you know, in any of the work I shared. So what am I working on now? These pictures were literally taken yesterday in my lab. So this is Jesse and Gianna. We are now looking at decomposition dynamics using something called the teabag index, if any of you are familiar with this. This is a standard protocol where you bury green tea and rooibos tea and basically see how much that tea decomposes under different experimental conditions. And the nice part about this is Lipton is very concerned with making sure all their tea bags weigh the same and have basically the same amount of tea in it. So great. That means my students don't have to do all of that precision work. Lipton has already done that for us, which is outstanding. So this is Gianna. We are making our current biochar out of sawdust. They were both very weirded out by the fact that I was like running around, jamming my phone in their face, taking photos. But this is currently happening. I have no data on this. This was taken yesterday. They are in the process of understanding bulk density and water-filled pore space in the soil, and how much biochar do we add? All of those calculations, they are in it. They are in it right now to try to understand how their work can help us understand a way to fight climate change. And so can old burnt coffee help us fight climate change? I would say yes. I would say yes. But I think more importantly, in many ways, it can help undergrads and young scientists buy into the idea that they have a voice in science. And it brings us opportunities to collaborate even more. So these are more pictures from when I brought my Marist students up here to carry. And so, for example, one collaboration that Jane and I have been chatting about, there are some dragnet plots up here. This is a disturbance experiment and nutrients trying to understand what happens to ecosystems, how they recover from disturbance, and how does that interact with nutrient additions. This is a very complicated figure just to tell you that this is sort of the broad experimental design of these what are called dragnet plots. And what if we, and these are the main goals. I literally just copied these from the Dragnet website. I did a cold copy and paste. I didn't ChatGPT it, I just copied and pasted it. But quantify community resistance and resilience after short-term nutrient addition. Does that change if we add biochar to those plots? Do the nutrients dynamic change? Do those dynamics change as well? Because biochar has been shown to be able to retain nutrients in the soil. Is that going to shift community? Is that going to shift composition? Is that going to shift all sorts of things? So why biochar plus Carry could work so well with scientists of all ages? I assume there aren't a bunch of high schoolers tuned into this, but maybe, I don't know what they do on the YouTubes these days. But just like I did with my undergrads, the great thing about biochar is the students get to feel it. They get to turn a lot of the control knobs and levers. They can choose what they're going to pyrolyze. They can choose how much they're going to add. They can choose the temperature and how long they're going to put that in the oven. They get to turn the knobs and dials. This is the power of biochar as a gateway. It's like a gateway drug into science, because they get to control stuff. They are when I talk to my students, say, what type of biochar do you want to make? They get very weirded out. They're like, what do you mean, do I want to make? No, you tell me what type of biochar you want to make. You get to make the decisions here. I could tell you, make biochar at 400 degrees for an hour, but you're the scientist here, right? So this becomes a mechanism for students, again, to be more engaged in science. And the nice thing is you can throw this biochar in a whole lot of conditions. As I said, dragnet under a lot of disturbance conditions, under different nutrient backgrounds. And the measurements that can be taken, the data that can be collected, can be as simple as plant productivity. How much biomass are these plants producing? What's the diversity of the ecosystems and the communities in response to this? Or we could answer more complex questions with carbon cycling, microbial respiration coming out of the soil, and decomposition. And this is just three. I mean, the list can go on and on. And so, as I said, right, the real power of biochar is the fact that it's this. It's something, it's a solution that anybody can hold in their hand. And I think this is also a way that, I used biochar when I taught at Poly Prep Country Day School in Brooklyn. It's a place, I had my 10th graders making biochar and looking at plant responses. I had my first year students here. I had my seniors. This is a place where students of all types can feel like they're engaging in real science and that they're not just following a procedure, right? If there's one thing I learned in all my years of teaching is one of the challenges we face as a community of scientists is we oftentimes, to our students, we sort of pull curiosity, we make them believe that science is just a set of steps for them to follow, right? But the interesting part of collaborative science is the fact that we don't know what's going to happen. So what do? We put our brains together and we say, hey, let's try this, let's try this. Jane and I were, this morning at breakfast, we were talking about the potential of building fake acorns that can be tracked with metal detectors? But both of us were like, I don't know, that seems weird, let's think about it, right? This is a tool that can be used where students can say, I made that decision. They're not just following a set of steps. And that's why I think it's a really powerful tool that can help bridge education, it can help bridge science, it can help bridge, oh, I can do science, right? Because decomposition, soil microbial, it's a weird thing. But, oh, I just put this in the soil and I see how it reacts, great. And I bring this up because ultimately, like this is a poll, this is some data from Marist students. Students are concerned about climate change. If any of you are familiar with Yale's 6 Americas, this is whatever this math adds up to, let's just round this up to 84% of students at Marist are either alarmed or concerned about climate change. They want to know how to deal with it. Science is the way to do it. So that's why I think this is such a powerful tool. And with that, oh, there it is. I'm happy to take any questions. Did I run too long?
We're at time.
Oh,
so what
all
I'm
right.
going to say is that please feel free, if you need to run, go ahead. But we will take at least one question here, and if you have to leave, go ahead. But otherwise, Ramesh will also be at lunch, so feel free to also stop him there. But does anybody have a fun or exciting, pressing question that they want to ask?
Yeah, Evan.
Go back to the only question, but I wanted to know why you picked 400 or 500 degrees. Like, why do you pick a number of biochar?
Yeah.
And what does that also mean for the amount of carbon converted into the biochar? Or is it just released to the atmosphere?
Right, right. The reason I picked 400 and 500.
Really quick. Oh, yeah. Will you rephrase it? Yeah.
Yeah, oh, sorry. So the question was, why did I pick 400 degrees or 500 degrees as the pyrolysis temperature to make biochar? Basically, it was logistical, because if you saw Gianna over there on the left, and all my other students made biochar by filling these basically soup cans with the feedstock. And if you put those aluminum cans, those tin cans, in really high temperatures, higher than 500, you get cans not surviving very well. And so it became it fully logistical. So that was really it, and 400 degrees was a high enough temperature where we saw some impact, and so then it also very simply became sort of a safety thing, right? How can we make sure students aren't sticking their hands in, they weren't sticking their hands in the oven, live streamers, okay, but how do we, it was a high enough temperature that we saw an effect, but that was really, it was more logistical than anything. I would love to do it at 1100 degrees, we just don't have the oven to do it.
Well, let's give one more big thank you, and for those of you who come.
Yeah.